a polymerase chain reaction, you'll need 6 basic things to complete the reaction. Your DNA template from whatever sample you're interested in, the primers specific to that template, DNTPs and polymerase, as well as buffer and molecular biological water to fill up the sample. All those things can be found throughout the lab. For now, the PCR tubes can be found over here in this cabinet. And there are these small, about tubes in this box, or box labeled like this. So all of the polymerase chain reaction materials can be found in the freezer. We have 3 different kits, Red Tack, One Tack, and Advantage. The Red Tack is special in that all you need to add is the template and primer specific to your sample, and already has the loading dye for a gel, so you don't need to add that. The Red Tack can be found in this box. All the files are right here. One Tack is found here, and Advantage is found here. Both of those have everything separate. The polymerase, the DNTPs are found here, and you can choose what you need based on the sample you're using. So I've pulled out the Advantage kit here. Each kit has its own specific manufacturer's instructions, and therefore the volumes for each reagent will change. But here, the Advantage kit fills up to 50 microliters total, with 1 microliter of polymerase, 1 microliter of each primer, both forward and reverse. So that's 2 microliters total. The primers can be found in the fridge, specific to your DNA template that you're interested in. Most of them will be on the shelf here in this metal box. The ones I'm using are found here. So I have the primers, the buffer, the polymerase, and the DNTPs. For this demonstration, I'm not going to be pulling out any volumes from here, but I just wanted to show you them, as well as I don't have a DNA template that I'm actually running through PCR, so I'll just be using water to fill up the sample. You also can create a master mix for the entire, if you're running multiple PCR tubes. For that, just multiply the volumes directed by the manufacturer by the amount of tubes you're going to create, and then possibly add 1 microliter more to those volumes, just because reagents can be lost due to transfer. So you're going to want to label your PCR tubes both on the top and on the side, just in case either one gets rubbed off, so you know which template is going in which slot, because sometimes you'll need to also run templates without DNA or without another reagent from your experiment, so you can have negative and positive controls. So here I've labeled both on the side and on the top. Once it's ready to go, you can start filling up with your reagents. And you're good to go. Once your PCR tubes are set up, you can head over to this machine, which is the thermocycler. You're going to have to turn it on, and the switch is in the back. It'll take a bit to power up, but in the meantime, you can go ahead and start loading. And you'll do that by untightening the top screw or latch, as well as lifting it up. Once you lift it up, you'll have all of these wells open to place PCR tubes, and you can just start loading it. And you can fill it whatever way you want, as long as you remember where you place the PCR tube specific for each template or for each reagent that you were using in your experiment. Once the system is finished booting, you can either enter a new protocol if that's necessary based on your PCR kit, but most PCR kits you already have entered into the system. So you can go ahead and select the Saved Files button. and there we have all the files here listed. So I was using Advantage, so I would use the Advantage underscore PCR kit, or PCR file. There are other ones, again, here for one-tack and other PCR protocols. Once you've selected your protocol, you'll need to close first the top, as well as tighten. And to tighten, you'll just want to use one finger until you experience some resistance. You don't want too tight, otherwise it'll cause problems for the reaction. Once that's all done, you can just hit run, and your reaction will go.